And thanks everyone for taking this webinar today between PacBio and Novogene Europe on long-read sequencing, innovation in genomics, harnessing HiFi sequencing and Kinnex solution for comprehensive insight. My name is Rehan Bai, Product Marketing Manager at Novogene Europe. And today we have special guests, Dr. Thea Ziegler and Dr. Dimitar Petrov from PacBio joining us. We are thrilled to invite you to this webinar as we dive into the applications and advancement of long-read sequencing technology. Let me start by a very brief introduction of Novogene. Founded in 2011, Novogene has grown into a leading global genomic services provider. Our state-of-the-art laboratory and sequencing facilities span multiple continents. With over 2,600 employees, including leading experts in genomics and bioinformatics, we committed to advancing genomic research. Here in Europe, our state-of-the-art laboratory located in Cambridge, UK, and Munich, Germany. The dedicated local team will provide you with comprehensive support and expertise to provide with exceptional data, exceptional efficiency and precision. And now let me take you through our journey with PacBio. So in 2017, we integrated PacBio SQL system. And in 2021, we upgraded to PacBio SQL 2e system. By 2024, we expanded globally and operating on 6 PacBio's latest sequencing platform, Review system, and 2 of which is located in our Munich lab. In the same year, we also launched PacBio Kinnex services, offering full-length 16S sequencing services and full-length RNA sequencing. In fact, just a few weeks ago, our Munich lab successfully tested the PacBio's latest Spark chemistry, obtaining great results. And we have now upgraded our workflow to Spark chemistry and provide exceptional data for our Europe customer. Dr. Thea Ziegler will tell you more about this exciting product later in the presentation. We are proud of our achievement with PacBio. Globally, we have processed more than 10,000 samples and contributed to the construction of reference genome for more than 1,000 species. We have run more than 4,700 PacBio Review Smart Cell since its launch. And our HiFi yield record is now at 150 gigabase HiFi data per cell with its latest Spark chemistry. In 2024 alone, we supported more than 150 publications, and the cumulative impact factor is now 1,070. The number will continue to grow. And lastly, let me briefly introduce our end-to-end PacBio sequencing services, including whole genome sequencing, full-length transcriptomic sequencing, and also metagenomic sequencing. So either you are building your reference genome, studying pangenome, or you are looking at variants, studying methylation patterns, or exploring isoform, study microbial metagenomics, we are here and we are happy to support your research. What's more, we also offer high molecular weight DNA extraction services in our Munich lab upon evaluation, and we also accept customer-built PacBio libraries, and we will perform review sequencing based on the latest Spark chemistry. And now let me introduce our speaker for today, Dr. Thea Ziegler, sequencing application cell scientist, a specialist from PacBio. Dr. Thea Ziegler has extensive experience in sequencing applications and will be our main speaker for today. We also have Dr. Dimitar Petrov from PacBio, and he will join us later in the Q&A section to provide with additional insight and answer your questions. So now, without further ado, let's get started. Please join me in welcome our speaker for today. Over to you, Thea. Stop share. Okay.
So, thank you, Ruyan. And also from my side, hello, everyone. Thanks for joining. I just quickly share my slides and then we'll start. So HiFi sequencing has played a crucial role in many of the genomic community's milestones of the past years. One significant example is its contribution to the completion of the human reference genome by the telomere-to-telomere consortium and the paradigm shift away from linear reference genomes toward pangenomes, which better capture the genetic variation across individuals. Thanks to significant innovations in throughput, ease of use, and the robustness of the neo sequencing platforms, the technology is used more and more in rare disease programs, such as genomic answers for children and all of us. In plant and animal research, HiFi sequencing has long been recognized as a platinum standard for generating new reference genomes. And beyond advancing our understanding of genomic complexity, HiFi sequencing also enables the comprehensive characterizations of epigenomes and transcriptomes. The unique combination of long read lengths and high accuracy is probably the most outstanding advantage of PacBio's sequencing technology. Beyond that, HiFi sequencing is free of systematic errors and delivers a uniform coverage across the genome, resulting in the highest possible genome completeness. It enables the sequencing of native DNA molecules, allowing direct single molecule analysis. This also permits the simultaneous detection of epigenetic modifications alongside standard 4-base sequencing. So how does this work? Sequencing is performed on a microchip known as SMART cell, which contains millions of tiny wells. Ideally, each well captures a single DNA molecule for the sequencing process. The detection of correctly integrated bases occurs via fluorescently-labeled nucleotides that emit tiny flashes of light when nucleotides are added to the replicated strand. Oh, sorry. Sometimes the reaction is a bit slow. So HiFi stands for high fidelity, referring to the exceptional accuracy. Today, the term is used synonymous with single molecule real-time or smart sequencing. HiFi reads consistently achieve 99.9% accuracy or higher, making the technology the most accurate long-read sequencing approach available. So what's the secret behind HiFi sequencing? The key principle is illustrated on the right. We call it circular consensus sequencing. To generate a circular DNA template, adapters are ligated to both ends of the double-stranded DNA molecule during library preparation. This allows a polymerase to make multiple passes around the same molecule during sequencing. Any DNA insert ranging from 500 base pairs to 20 kilobases can be sequenced. And thanks to the random distribution of errors, the system can compute a highly accurate consensus sequence from the multiple subreads. This final sequence is what we call a HiFi read. PacBio provides products and support for the entire workflow, from sample preparation to analysis. For the critical step of high molecular weight DNA extraction, our nanobind kits offers optimized protocols for a variety of samples. For library preparation, both manual and automation-friendly high-throughput solutions are available. For sequencing, we offer different sequencing systems. The review system that you can find at Novogene is our most powerful system. Thanks to the onboard processing of collected data, the resulting output file is very compact, reducing storage requirements. Our free SmartLink software offers comprehensive solutions for primary and secondary analysis. The integrated tools for the characterization of specific variants are an important highlight for many users. The extraction of high molecular weight DNA is one of the most critical steps in the workflow. Our nanobank kits are specifically optimized for HiFi sequencing and support a wide range of samples. The SRE kits can further improve overall yield by removing shorter DNA fragments, which is especially important for sequencing larger genomes or multiple samples. It is worth putting some effort in the optimization of high molecular weight DNA purification, as this will affect the quality and yield of the data. PacBio recently introduced a new chemistry for its review sequences, which has excited our users due to significant advantages. First of all, the reduced amount of required input DNA, requiring 4 times less DNA compared to the old chemistry. At the same time, the new SPARC chemistry offers an increased output, lowering overall sequencing costs. Also, an instrument and methylation calling has been expanded with an additional methylation marker, simplifying the analysis of 6-MA. In addition, 5-MC calling accuracy has been improved by 10%. Novogene was among the first users who implemented our new chemistry, so that you will benefit from all of these advantages when using their services. The 4 times lower input enabled by the new SPARC chemistry reduces the required input DNA amount down to just 500 nanogram. This enables applications with previously inaccessible samples, such as saliva or precious biobank samples, where only limited materials are available. In addition, we see a significantly increased output of HiFi data. With one smart cell whole genome sequencing run, we now get 120 gigabases with the new SPARC chemistry. Thanks to improvements on the analysis side, the variant calling performance for 20-fold versus 30-fold HiFi genome coverage is nowadays actually quite small. The figure on the left, for each of the 3 different variant classes, the F1 accuracy at 10-fold, 20-fold, and 30-fold HiFi coverage is plotted. So if you look at the difference between the accuracy at 20x versus 30x, it's actually very small. But there's obviously a significant cost advantage when using 20x coverage, as it will enable you to sequence 2 human genomes on 1 single smart cell. So this has just become a reality now, sequencing 2 human genomes on 1 smart cell, which halves the price compared to before without SPARC. Regarding our multi-omic and epigenetic detection capabilities, our SPARC chemistry provides improved accuracy and increased confidence in 5-MC calling. This results in significantly improved cost efficiency for methylation studies. For human samples, up to 8 samples can be processed in a single smart cell, enabling large-scale studies with comprehensive insight into which regions of the genome are methylated and which are not, now at a very attractive pricing. Additionally, we have integrated a 6-MA caller for multi-omic fiber-seq chromatin assays directly on the instrument. As known from other NGS technologies, our HiFi applications cover a broad spectrum of applications. During the remaining time, we'll focus primarily on the genome assembly examples from plant and animal, and also RNA sequencing and the improvements in this area thanks to our new Kinnex kits. And finally, we'll have a look into our microbial applications. HiFi sequencing is particularly powerful for tackling large, polyploid, and highly repetitive genomes because it overcomes key challenges that traditional short-read sequencing struggles with. There are numerous examples where HiFi sequencing was used to successfully resolve even very large and complex genomes. We will see some examples in a bit. The reasons why HiFi reads are so useful for resolving complex genomes are, first of all, the long read lengths that allows them to span repetitive regions. With lengths up to 25 kilobases, they are able to bridge all of the shorter repeat regions of the genome, while the high accuracy permits the differentiation between copies of long repeats. And in genetically diverse or output populations, haplotype resolution is critical for understanding allelic variation. With 99% accuracy or higher, HiFi sequencing enables phasing of all haplotypes, regardless of the ploidy level or complexity and size of the genome. So here we see an example of the hexaploid persimmon genome. The data was kindly provided by Jeremy Schmatz from Hudson Alpha. It was still obtained with the previous radiochemistry version, which yielded around 90 gigabases per run. So 3 smart cells were required for the 6 times 800 megabases, yielding 267 gigabases in total. You can see very nicely in the IGV plot on the right that all 6 haplotypes of the persimmon plant could be phased, based on the distinction of unique SNPs. Each sequencing project is unique and varies in the given conditions. While some genomes present with their complexity and size, others are so small that the main difficulty lies in isolating sufficient high molecular weight DNA. Or for some species, no reference genome exists yet, but HiFi sequencing makes de novo assembly straightforward, producing genomes with reference genome quality. To accommodate different needs, we offer various protocols for whole genome sequencing. For projects with very limited amounts of input DNA, we offer an ultra-low input for workflow, in the newest version called Amplify. This approach is amplification-based and requires as little as 1 to 15 nanogram of DNA input. The protocol has been updated and optimized with the new SPARC chemistry, now enabling genome sequencing for genomes up to 3 gigabases. Recently, research teams from Hudson Alpha and the University of Georgia developed a new log coverage trait mapping workflow. This study highlights the power of low-pass HiFi sequencing as a cost-effective alternative to short reads for high-resolution trait mapping. The results of the study showed that at equivalent coverage, long reads far exceed short read sequencing in confidentially calling SNPs, identifying high-value genetic variants, and capturing the broader variant landscape. At 1.6-fold coverage, HiFi sequencing still covered 55% of the genome and 58% of the gene space, compared to 17 and 11% with short reads. This resulted consistently in higher locus similarity scores for important disease resistance loci, for example, for late leaf spots. The authors estimated that using HiFi reads resulted in around 8.5-fold decrease in cost per value compared to short reads. They concluded that their findings position long-read sequencing as a more economical choice for high-resolution genomic analysis, particularly in projects requiring extensive coverage of complex genomes. As already mentioned earlier, our review sequencers are able to detect 5-MC and 6-MA methylation markers in parallel to standard 4-base sequencing. The methylation calling is integrated to the instrument, turning our sequencers into real 6-base sequencers. As HiFi reads achieve higher genome completeness than short reads, nearly from telomere to telomere, you'll be able to access 13% more of the genome than with bisulfite approaches. Having long and accurate reads with additional insights about methylation markers allows you to look at very interesting biological questions. Instead of seeing the methylation profile of collapsed haplotypes, you'll be able to separate the maternal and paternal haplotypes or all chromosomal copies that are there, and have a deeper look into epigenetic regulation mechanisms in development and disease. Here we see the methylation profile of a genomic region surrounding the SRF gene in a human sample. The SRF gene is known to play an important role in gene regulation in vertebrates. As you can see, the transcription start site of active genes are hypomethylated, shown here in blue. The results are consistent with findings from bisulfite sequencing and EM sequencing approaches. Now we're looking at the autologous genomic locus on chicken. And again, we find the transcription start sites of the SRF gene and further activated genes hypomethylated, proving the applicability of our 5MC tool in other vertebrate species. In case you're asking yourself about the value of the 5MC calling tool in the context of plant research, here an example from Michael Schatz's group, who tested it on tomato and mice. Plants contain lots of transposable element regions and cannot be mapped with short reads. Including them to your genetic analysis would allow you to look at interesting questions, for example, studying how methylation controls transposable element silencing in crops like maize and wheat, or how it affects genome stability and adaptation. The shown results were presented at a former AGPT Ag conference, and as you can see on the right, the methylation profiles from PacBio sequencing were highly correlated with bisulfite sequencing. The gaps that you see at the bisulfite sequencing trace, marked with the red arrows, comes from transposable element stretches that are not covered. So HiFi sequencing was able to yield additional insights. The conclusion from genomic expert Mike Schatz was that the methylation levels from HiFi are highly correlated with bisulfite sequencing, and HiFi long reads make transposable element methylation measurable. In mice, the transposable elements make up 80% of the genome. So far, the highly repetitive nature of transposable elements has hindered our understanding of their regulatory potential. In a recent study, a team led by Andrew Stergachis from the University of Washington applied long-read chromatin fiber sequencing, brief fiber-seq, to mice. Fiber-seq is a chromatin profiling method developed by their lab that marks open chromatin regions via enzymatic 6-MA methylation. Applying fiber-seq to mice enabled comprehensive annotation of the regulatory potential of transposable elements, revealing more than twice as many accessible chromatin regions compared to ATAC-seq. As classical NGS technologies, HiFi sequencing is compatible with targeted sequencing methods. The two most common approaches are depicted here. Due to the longer read lengths, the complexity and effort of primer or probe design is significantly reduced in both cases. Typical amplicon lengths for HiFi sequencing are up to 10 kilobases, but there are also cases where users have gone further. Ultimately, anything that can be amplified can be sequenced. Long-read amplicon sequencing is a method of choice for those interested in single gene or small gene panels, and for larger panels, as a rule of thumb, for more than 10 to 12 target genes, we recommend target enrichment as a method of choice. As a suitable partner, we can recommend Twist Bioscience. They were the first to optimize the hybridization protocol specifically for HiFi sequencing. Both amplicon sequencing and target enrichment are well established and provide robust results in combination with HiFi sequencing, even for regions that are not accessible with short reads, with the well-known advantages, such as the resolution of different haplotypes. Another targeted sequencing solution is our new Pure Target Kit, which provides an effective approach for repeat expansion sequencing, a challenge that is traditionally very difficult to resolve with short reads. There are many repeat expansion genes involved in neurological diseases, and determining the exact length is important for distinguishing between healthy and pathogenic variants. For this reason, PacBio has compiled a panel for the 20 most important repeat expansion genes, including many ataxia genes and Fragile X. The target enrichment approach is CRISPR-Cas-based, enabling insights of the methylation profiles. The Pure Target Repeat Expansion panel provides a convenient end-to-end solution for repeat expansion analysis, delivering answers for previously unresolvable genomic regions in just 3 days. Here you get a glimpse what the Pure Target data looks like, analyzed and visualized with our target tool. It will allow you to obtain comprehensive insights about base resolved repeat sequence at a single nucleotide level, such as the example in the upper left, but it also provides exact sizes of repeat expansions for both alleles. We'll also provide information about the methylation status, as the assay involves the work with native DNA, and the high read depth even allows to resolve mosaicism. Before we transition to our RNA sequencing applications, I briefly wanted to highlight our innovative data analysis tools, which is another important differentiator. Most of them were developed by our own team, with the exception of the deep variant tool, all above. This empowers our users to analyze very challenging variants, providing more discovery power than any competing technology. One example is the target tool that you've already seen for the detection of tandem repeats. In tandem repeats, it spans 3.8% of the genome, so it's not a neglectable portion. And paraphase is another example for the detection of segmented application, which are traditionally very difficult to resolve with short read. This variant class spans 7% of the genome and makes up a large portion of the formerly dark region of the genome. And for methylation analysis, there's our MethBAS tool, or rather a collection of tools for 5-MC methylation analysis that enables, for example, the creation of cohort profiles from a collection of methylation profiles. As we're all likely familiar with, when DNA is transcribed into RNA, it can undergo alternative splicing, which generates multiple mRNA isoforms, each potentially present at different abundances. This greatly increases the protein coding potential of the genome, but it also makes it challenging to characterize the transcriptome. In recent years, it has been repeatedly shown that the importance of alternative splicing has long been underestimated. Since different isoforms lead to different proteins, which represent the carriers of function, it is important to be able to make an accurate distinction. While short-read sequencing can only assemble 20 to 40% of the human transcriptome, PacBio's isoseq method offers a complete view of isoforms, as it sequences each transcript in one single read, however, on the cDNA level. The ENCODE study was the first large-scale cross-species survey of transcript structure diversity using full-length cDNA sequencing on long-read platforms. Full-length transcripts detected accounted for around 88% of annotated human protein coding genes, identifying 200,000 full-length transcripts, of which 40% contained novel exon junctions. The key findings from the study were that transcript isoform diversity is more complex than previously thought. More than one predominant transcript was found for 73% of genes, contradicting former reports suggesting single dominant isoforms. Also, transcript structure diversity and major transcript usage vary significantly between tissues, samples, and developmental stage. One big surprise was that across human and mouse, the dominant source of transcript diversity differed for more than half of autologous protein coding genes. This challenges the widely used practice of extrapolating human gene function from mouse models. Another important application of our isoseq method is genome annotation. In the example shown here, the method was used for genome annotation in an ant species. The researchers of the study highlighted two key aspects. First of all, the correction of misannotated genes, shown on the left. The researchers noted that isoseq helped to correct many incorrectly annotated genes. In the specific case shown, two previously separated gene models were found to actually belong to one single gene. And the second important finding was that improved genome annotation enhances other NGS data sets, as shown on the right. In this case, with a new improved isoseq-based annotation, 44% more of the 10x Genomics single-cell RNA sequencing data set could be mapped to the genome, significantly improving data utility and biological insights. Therefore, better genome annotation can make other data sets, even short-read data, more valuable. So isoseq has been a valuable tool for researchers to distinguish and characterize isoforms for a long time. However, the method has not been very cost efficient in the past, which has limited its use. This is because the average transcript length is between 1 and 3 kilobases, while the optimal length of a HiFi read is 15 to 20 kilobases. To solve this problem, researchers at the Broad Institute developed a method a few years ago that concatenates transcripts or transcript cDNAs into fragments of an optimal length for HiFi sequencing, so between 15 and 20 kilobases. The method is very robust and provides the same sequencing quality as a classical isoseq method, since all cDNAs are labeled with unique barcodes and delinked into the individual transcript cDNAs in the end. Based on this method, PacBio developed the Kinnex kits for cost-effective scalable RNA sequencing. For full-length RNA sequencing, the Kinnex approach enables a 12-fold concatenation, yielding up to 60 million reads on the review system in combination with the Spark chemistry. For this reason, RNA sequencing with PacBio is more affordable than ever before. And when you consider that a long read provides significantly more information than a short read, the cost per value is probably smaller. The Kinnex kits also exist for single-cell RNA experiments and for 16S amplicon sequencing. For the first time, the new Kinnex kit provides the throughput needed for large-scale multi-sample studies. Our team tested different cell lines and tissues to determine the read depth required for isoform discovery saturation. They found that most of the moderately to well-expressed transcripts can be saturated and quantified at around 10 million reads per sample. And also that isoform diversity yields approximately 1 new isoform discovered for every 1,000 reads. So this makes the discovery of new isoforms and associated regulation mechanisms highly likely when trying Kinnex. This table provides a summary of our sequencing recommendations for different applications. For isoform discovering comparison of moderate to rare transcripts, we recommend a read depth of 10 million, which allows you to sequence 6 transcriptomes in parallel on a review. For isoform discovery of highly expressed transcripts, you can reduce read depths down to 5 million, which increases the number of samples that you can sequence in parallel to 12. For comprehensive representation of expressed isoforms in plant and animal research, the choice of the read depth depends on the complexity of the species transcriptome. We recommend to start with a sequencing depth between 5 and 10 million reads. Here you see a scheme about the experimental setup. For Kinnex full-length RNA experiment, you will need around 300 nanogram of total RNA as an input with a RIN/7. And the provided barcodes allow you to multiplex up to 12 samples or tissues in parallel. The library prep takes around 2 days and sequencing less than a day. The read segmentation and isoform classification are supported by our SmartLink software. For single-cell experiments, the Kinnex protocol starts with the created single-cell cDNA library. The Kinnex single-cell RNA kit is compatible with the 10x chromium 3' kit and 5' kit, and since recently also with the PARS EverCode kits. As an input, 15 to 75 nanogram cDNA are required. The achievable read number is sufficient for single-cell experiments of 3,000 to 10,000 target cells. Our barcoded Kinnex adapters support 4-plex multiplexing, so you can pool several single-cell experiments per smart cell if wished. And after the primary analysis with our SmartLink software, the resulting output files are compatible with classical tertiary single-cell analysis tools. For example, Zurat, Scampi, Kanna. The big advantage of single-cell sequencing with PacBio is that thanks to the high accuracy, cell barcodes and EMAs can be resolved as good as with short reads. And the data is therefore fully usable, unlike with noisier long reads. And besides that, Kinnex single-cell reads offer additional valuable isoform level information missed by short reads. Remember that short reads usually only sequence around 50 base pairs of the 3' end in single-cell experiments. This means that all possible isoforms are assigned to a single occurrence of the gene, assumed to be the dominant form. However, studies such as ENCODE 4 have shown that this assumption is often not correct. So finally, we now come to our microbial applications. There are 4 primary ways you can use PacBio HiFi sequencing to understand your microbial community and microbial species. For whole genome sequencing, you can complete microbial genomes with ease and confidence. For most microbes, closed genomes with accessory plasmids can be assembled with one touch using the default settings of our assembly pipeline. Short-read polishing is not needed. For full-length 16S sequencing, is employed to reveal the true diversity of your samples with better taxonomic resolution and generates more testable hypotheses. We'll have a deeper look to that in a minute. HiFi shotgun metagenome profiling provides more and richer functional information, yielding on average 8 genes per sequence, with 90% of reads containing at least 1 gene. Metagenome assembly provide far more high-quality max than other technologies and more complete circular and single-condig max, even at lower coverage. Short-read polishing or hybrid assemble is again not needed with HiFi. Our HiFi accuracy usually exceeds the accuracy of short reads. So we're probably all aware of the usefulness of 16S sequencing, but what's the value of full-length 16S sequencing? The complete 16S gene, including the variable regions V1 to V9, gives a much better taxonomic resolution than short reads, which usually only target 1 or 2 variable regions. In contrast to noisier long reads, thanks to its superior accuracy, HiFi reads are able to distinguish key species and strains, which can be critical in some cases. For example, when differentiating pathogenic strains from commensals. Other full-length amplicons like fungal ITS or eukaryote 18S benefit as well from better taxonomic resolution with HiFi. You just need to order the region-specific primers. The bacteria species are highly similar and difficult to distinguish with partial 16S sequencing. Short reads only target a specific region and can miss intergenomic SNPs, which may be present on other regions of the gene. With full-length sequencing, you will be able to pick up all intergenomic variants for strain-level resolution. The shown figure shows intragenomic 16S gene polymorphisms in human gut microbiome isolates. The information required to do strain-level assignment is present in most of the human microbiome project isolates, but the location of the variants vary by bacterial clade. So in the rectangle, you see that these are the variable regions that are picked in most short read approaches. In contrast, long reads cover the complete 16S operon, and therefore full-length 16S sequencing will be required in order to obtain strain-level resolution by leveraging all of the intragenomic variants. For metagenome assemblies, the goal typically is to assemble a representative genome for each microorganism in a microbiome sample. However, there are several challenges that only accurate long reads can resolve. For example, there are multi-species present, leading to uneven and unknown coverages, and there are many repeat regions and intergenomic overlaps across species and strains that pose a hurdle that short reads usually fail to resolve. In contrast, thanks to the combined long read lengths and high accuracy of HiFi reads, they allow to assemble a large number of high-quality MACs in a single metagenomic shotgun experiment. So here we see an example data set of a pooled human gut microbiome sample from around 35 million HiFi reads and 260 gigabase data in total. HiFi as a meter was used for the assembly and produced 75 high-quality MACs as closed circular contigs. As you see, they comprise smaller and larger microbial genomes from 1 to 6 megabases. Yeah, so this is how it looks. It's pretty impressive, right? The metagenomic profiling is also a good way to investigate viral samples. The shown data comes from a study conducted by PacBio scientists. They performed a deep sequencing experiment with PacBio HiFi reads to obtain metagenome assembled genomes from a pooled human gut microbiome. For long read metagenome assemble, they used two methods, HiFi as a meter and meet-MDBG. Both methods had their advantages. So more viral sequences could be detected in the meter-MDBG assembly, while the HiFi as a meter assembly detected more pro-viral sequences. Overall, the improved metagenome assembly methods and complementary binning strategies were highly effective for rapid cataloging of complex microbial populations, including viruses and pro-virus plasmids from microbiomes. So throughout this presentation, we have seen how HiFi sequencing has been a driving force behind numerous advancements in the genomic field, providing a more complete view of biology. Thanks to its long and highly accurate reads, HiFi sequencing enables the generation of high-quality telomere-to-telomere genomes and pangenomes, capturing structural variation across individuals. It also permits the simultaneous epigenome analysis, so direct detection of 5-MC and 6-MA methylation without additional library preparation. It also enables the generation of isoform-resolved transcriptomes, enhanced with Kinnex kits offering higher throughput and cost efficiency. And last but not least, the value for characterizing complex microbial populations using 16S Kinnex or metagenome profiling for in-depth microbiome studies. Yeah. With that, we're coming in to an end and are open for questions.
Thank you, Thea. It's an amazing presentation. And during the presentation, we received a lot of interesting questions from our attendee. So the first question is, what is the error regarding the PacBio? Is it compared to other long-read technologies to corroborate the data?
Okay, hi everyone, and probably I can elaborate a bit on that. So the strength of PacBio sequencing is just it's better quality or accuracy, which is achieved by these multiple passes over the same molecule. And what we usually see with Kinnex, for example, so concatenated fragments, or let's say lengths of fragments of 15 kb or more, is usually quality scores of read qualities of in the range of, let's say, Q33, 35. So that's usually better than other long read technologies to simply answer the question. So yes, the simple answer is the quality, read quality, and also base quality is usually higher. I would say significantly higher, and it could be also improved if your fragment lengths are shorter. So if you are doing iso-seq, this means without concatenation. Of course, you have shorter fragments, and in the time period of sequencing, you can generate more passes of the same molecule. So the quality scores will be also significantly higher as well. So that's the short answer.
Thank you, Dimitar. And the second question is, for targeting gene enrichment, do you provide probe design?
Oh yeah, Thea, do you want to answer?
Yeah, so it's a simple answer. The answer is no, but we do offer a shared protocol together with Twist Bioscience, so you can turn to them for target enrichment panels, and they will provide support with the probe design.
And for the pure target kit, which we have, which is designed for 20 targets, which doesn't involve amplification. If, for example, you're interested in methylation, we don't guarantee, let's say, success, because it's always depending on if you design your probes, but we can discuss how you can approach this. So if you are planning this kind of project, probably you can directly contact us, like PacBio or Novogene, and they can forward basically your message to us. So we can discuss basically, it is possible also for the pure target, PacBio kit which is no amplification kit for target enrichment, to design your own targets or your own probes in that sense. But yeah, I mean, we have optimized it for 20 targets. If you need more targets, of course, this is more like something you should optimize, but we can provide you with some guidance.
Thank you both. And the next question is, are there any optimization of the 10x workflow to better capture longer RNA molecules before moving into Kinnex, increasing reverse transcript time, for instance?
Yeah, that's another good question, which we've heard a few times as well. If you're interested in longer transcripts, more specifically, 10x usually gives you a rather shorter range, let's say, or let's say the distribution of fragments is more in the range of 1 to 2 kb, whereas for longer transcripts, what also 10x themselves actually also recommend, and what we've also seen from our customers, is to increase the elongation times during cDNA synthesis. This can give you a bit longer or gives you then a bit longer, more of the longer transcripts, let's say. So this is something you can do. During concatenation itself, we have not observed any significant change in let's say, or bias towards shorter fragments. But yeah, this is, you can increase also during the Kinnex kit the elongation times if you are aiming for transcripts above 5 kb, for example.
Thank you, Dimitar. And the next question is, do you have a freely available pipeline, for example, on GitHub for alignment, variant calling, fixing, etc.?
Yeah, that's I believe actually it was mentioned during the talk, but it is the case. So we have a good GitHub page where we have a lot of pipelines which are freely available. Our software is also free, so our smart link software, which you can use for click and play basically analysis, so not command line analysis of your data. But on our GitHub repositories, we have all the so we have more options for analysis, and all those which were mentioned, we have like phasing or variant calling and so on. We have our pipelines and you can use them. We'll need to paste somewhere the links for that, of course, or you can Google, of course, just PacBio GitHub and or analysis, you'll find it actually.
And the next question is about iso-seq. So is there a difference between iso-seq non-concatenated and Kinnex results?
Yes, it's a good question. So iso-seq is the non-concatenated options, just cDNA directly as you extracted the RNA, and then Kinnex the concatenated option. There is no, like, we have sequenced basically the same library before and after concatenation, and we see the same results. So we don't see any bias caused by the Kinnex formation, so by the concatenation.
Okay, thank you. And do I need an additional library preparation kit for Kinnex?
Yeah, that's not the case. So the Kinnex kit is enough for, like, it is doing concatenation and at the same time ligating the adapter, so preparing the DNA or cDNA in this case for sequencing. So you have directly your ready library at the end. So everything, concatenation and library prep, is done in one step.
Okay, thank you. And the last question is, can I use a flexible concatenation factors?
We have 3 kits which have different concatenation factors. It's 8, 12, and 16. The aim of the concatenation factor is that you are in the range of 15 to 20 kb, which is our ideal size, let's say. So this is how you can choose the concatenation factor. Of course, each kit comes with a specific concatenation factor because, for example, 16S Kinnex, the 16S Kinnex is 16S size is 1.5. So therefore, the range there ideally would be 12 in factor. But you can, so the kit is made in that way that you can exchange concatenation factors. So if you take the first bit, for example, of the single cell, which is basically treating the cDNAs in preparing it for concatenation, then you can take for the second part, for the actual concatenation, the, for example, 12 or whatever. I mean, if it's single cell, of course, probably, I mean, in that case, you should not, I mean, it makes no sense to change the concatenation factor. But if you have amplicons, for example, where you want to change the concatenation factor because the size is varying, then this is possible, yes. But of course, it's not officially supported. I mean, we have tested those kits and these options which we sell as a ready-to-use kit, but potentially, yeah, it's possible, technically possible, to change the factors.
Thank you very much, and that's very helpful. And thank you all for your time, and we hope you have enjoyed this webinar and found it useful. And thanks for all your questions. As mentioned, if we haven't managed to answer your question live, we will get back to you via email next week with the answer. We will email you with the link to the webinar recording and the slide deck. You can expect to receive the week after. When the webinar finishes, a survey will pop up, and we'd appreciate you to take the time and complete it as it will help us to ensure we make future webinars even more relevant to you and your research goals. As a token of our appreciation to your attendance today, Novogene is pleased to offer you a 15% discount on your upcoming project. An email will be sent to you within the next few days with a code that you can use to claim the discount. There will be a link in the follow-up email for you to view a certificate of attendance if you need one. And this can also be printed from your browser window. In the meantime, you can also follow us on X and LinkedIn to keep updated with the future webinars, offer, and news. We look forward to seeing you on our next webinar. And thanks again for everyone. Thank you. Have a nice day.
Thank you.